Thank you. It's quite an honor to be here. A tremendous thank you to Alex and the team at Groundswell for inviting me. This is my first trip. It's the first time I've ever been in the UK. And I think only the second time I've been east of the Atlantic Ocean, or maybe the third. So thank you for the warm welcome. I was quite privileged to arrive here on Monday, and I spent the day yesterday visiting with some farmers and some growers, developing an understanding of the farming context, the climate, the environment, and I've had many friends that I've spoken with here for a number of years. And the topic, the framework that I want to speak a bit this morning is the opportunity that exists, the unrealized potential. And if you've listened to the podcast, you've listened to some of our webinars, you've certainly heard me speak about this topic before, although I haven't as much recently. But I think many of us don't really have a concept or an appreciation for what remarkably healthy plants can actually do, the yield potential that actually exists, the disease and insect resistance potential that exists, simply because most of us have never observed it. And most of us have never observed and really understood the agronomic management principles that need to be put in place to make it happen. So the discussion this morning, I want to focus on a very high level of this is the opportunity that exists. This are some of the pieces that need to be in place to make it happen. And then the discussions later today and tomorrow, I want to dive much more deeply into the practical applications of this is actually how you do it. This is how you put these ideas and concepts into practice and how you make them work. So Alex preempted me with the question. I wanted to understand how many of you have listened to the podcast, so thank you, Alex, for that. Instead, I'm going to ask a different question, slightly different question. On the podcast, webinars, or on whatever platform, how many of you have heard me describe and relate my personal story, the farm that I grew up on, and how I came to be in this group, okay? A number of you did, but most of you did not. I think this is important context. So, I grew up on a family fruit and vegetable farm in northeast Ohio, in the snow belt south of Lake Erie, so I'm about 30 miles south of Lake Erie. Historically, we used to get 90 inches of snowfall annually, and that hasn't happened a lot in the last decade. We typically receive 40 inches of total accumulation, some of that as snowfall, and some as rain, 40 inches rainfall per year. We are the second cloudiest spot in the continental 48, second only to Seattle, in as high as 280 days of cloud cover per year, and very high humidity. 80 to 95% humidity is common during the summer months. So when you think about that, that is the perfect petri dish environment for propagating disease. I grew up on a family fruit and vegetable farm, a small farm. We had 25 acres growing a combination of different fresh market vegetable crops. Tomatoes, zucchini, cucumbers, and cantaloupe were the four primary crops that represented about 70% of our total production. My father was also a local supplier of seeds, fertilizers, inputs, and pesticides. He was also the pesticide distributor for the local region, and as a result, we were the first in the community or in the region to test and evaluate new pesticide products that were coming to the marketplace, and then make recommendations to our growers on how well they were working for us. In the early 2000s, we had a 3-year consecutive period, 2002, 2003, 2004 growing season, when we lost upwards of 70% of our 4 primary crops to a variety of different diseases and insects that we were unsuccessful in managing with pesticide applications. We were dealing with powdery mildew and downy mildew on our cucurbit crops, squash bugs, cucumber beetles. On the tomatoes, it was septoria leaf spot, early blight, and late blight. And it seemed the more intense pesticides that we applied, the greater the problems became, until we had this 3-year consecutive period. We had started growing vegetables in 1994. Now here we are a decade later and having tremendous significant crop losses. And then in the 2004 growing season, the 3rd year of this 3-year period, we had a wake-up moment, what I would call a 2x4 in the face kind of moment, where we started renting a field from a neighboring farm that bordered right up against one of the fields that we've been farming for the prior decade. And so these two fields used to be long, narrow strips that we were tilling and planting up and down the slope because of how narrow they were. Now that we were farming both fields, we switched the road direction 90 degrees and started planting across the former field border. And in the section closest to the road, of course, where everyone could see it who was driving by, we planted cantaloupe. And on the soil that had the previous decade of intense pesticide applications, even though we were doing cover crops during the winter months, incorporating compost, we were doing many things well, the plants on that soil at harvest time had 80% of their leaves infected with powdery mildew. And on the new soil, there were literally plants that were planted 2 feet apart. On that new soil, the plants had no powdery mildew. And when I say no powdery mildew, I mean zero. There was not 5% or 10% infection. We couldn't find powdery mildew in that field. There was this very clean knife line right down where the former field border had been. And this boundary was so pronounced that there were healthy vines growing right in among the unhealthy vines. And that, as you might imagine, got our attention. I wanted to know what are the differences between these two plants and what allows one plant to be resistant to powdery mildew when the next plant 2 feet away is susceptible. And thanks to some remarkable mentors and a lot of reading and research and discovery, over the next several months and following years, I learned that plants have the capacity to be completely resistant to all diseases and all insects as long as their immune system is supported with nutrition and with a foundational microbiome. We understand that all of us have our own immune system, but they don't all work equally well. Some people become ill with the first cold or flu bug that comes along, and other people practically never become ill. And the only reason for the difference between those two is how well their immune system has been supported with nutrition and with microbiome health from even before they were born. And the same concept also holds true for plants. Plants have 2 different immune pathways. It's too much to get into for the moment, but conceptually, it is possible to grow plants in such a way that they're completely resistant to diseases and insects, and by the way, it is not that difficult. It's different, but it's not difficult. One of the pieces that has so intrigued me is how we have been very fortunate to have had some very good mentors and guidance and to be able to work with an amazing group of farmers. Alex mentioned that today we're working on about 4 million acres, and that represents about 55 different crops that we have in-depth data and extensive experience with. That ranges from tree fruit to nuts to grapes, all kinds of vegetable crops, and then of course broadacre crops, small grains, corn, wheat, beans, etc. And every year for the last decade and a half, a group of growers from somewhere in North America or around the world will approach us and say, we have this new emerging pest, this new emerging disease for which there is no known treatment. There is no effective pesticide or fungicide that will control this disease. Do you think you can solve it? As of this point, we haven't failed once. We have a perfect track record of solving every one of those diseases. But if you were to ask me, all right, what is the solution for bacterial canker on cherries? I don't know the answer. I don't have a single simple point answer that says, well, you need to add more cobalt and zinc and manganese and you'll solve bacterial canker on cherries. Because the approach that we take is a very systemic approach to evaluate overall soil health, soil microbial function, and plant nutritional balance. We just address all the bases. We take care of the foundational aspects, and we look at plant nutrition through this very different lens, and the problems disappear. So when we consider plant nutrition through this different framework, agronomy of the last century has been framed around balancing plant nutrition with one objective in mind, and only one objective, and that's to produce the highest yields possible. Our approach is the same with a twist. And the different perspective is we want healthy plants that produce the highest yields possible and that have a functional immune system and are resistant to diseases and insects. And here's the fascinating piece. When you produce plants that have such robust immune function that they're resistant to diseases and insects, not only does that eliminate the need for pesticides, but also when you have abundantly healthy plants, you can't stop yields from increasing. And there is this idea that there are now many voices, at least in the North American regenerative agriculture space, who are now speaking out against this idea. There has been this prevailing idea that as we begin this transition to a regenerative agriculture system, that we should necessarily expect to see yield declines. We should expect to see yields go down because of increasing disease pressure, increasing insect pressure, and increasing weed pressure. That has been the opposite of our experience. Our experience has been that when you manage agronomy well, when you manage plant nutrition well, you should expect to see the opposite results. We expect to see yields go up. How can you have abundantly healthy plants and expect the yields to go down? That makes no sense. But the key difference is that our approach to facilitating a transition is not to just remove things. We don't just remove pesticides and call that regenerative agriculture. We need to earn the right to reduce synthetic inputs. Our crops need to earn the right to reduce synthetic inputs. We need to, before we can have a conscientious conversation about reducing pesticide applications, we first need to increase plant health to the point where they're not required, where they're not needed at all. The foundation for this, one of the foundational aspects of this conversation came about when as we transitioned on the farm. I didn't finish the story of what happened on the farm at home, but we went through a very rapid transition in the 2005-2006 growing seasons. By 2006, we were completely pesticide-free. And I noticed some I came to several fascinating realizations that when you have plants that have this robust immune function and are completely resistant to diseases and insects, not only do they have their own inherent immunity, but they have the capacity to transfer that immunity to people and to livestock. And we can have a legitimate conversation about growing food as medicine. Secondly, we observed that when we're growing these remarkably healthy plants, we are also rapidly regenerating soil health while we are growing a crop. And this is an important clarification. We've had the paradigm collectively for the last several decades, I think for the last century or more, that agriculture is somehow, by its very nature, inherently extractive, that the process of growing a crop is going to remove nutrients and remove organic matter from the soil, and that if we want to regenerate our soil health, we will then need to replenish that with cover crops, with green manure, with manure and compost applications, with carbon applications, that we somehow need to then import things to regenerate the soil, to replace what we've removed with crop production. It's true that this is common today. Many plants, the way that many crops are managed today, they do deplete soil. But it's not necessarily true. It's not the default status. It is possible for plants, when you go back into the agronomic literature about 5 to 6 decades, during the 40s, 50s, 60s, even into the 70s, there were many leading agronomic articles of the day, peer-reviewed papers, and in the popular literature, who described that the fastest way to build soil was to grow corn. Today we have the idea that the fastest way to deplete soil is to grow corn. So what has changed and what is different? What is different is that what has changed is, as a result of mineral nutrition depletion and microbiome depletion, we have plants today that no longer photosynthesize efficiently, that no longer photosynthesize well. What we perceive as common and normal is plants that photosynthesize somewhere in the neighborhood of about 15 to 20% of their inherent photosynthetic efficiency. 15 to 20%. Now, achieving 100% in an outdoor agricultural environment is unrealistic, because to achieve that would require a perfect environmental conditions, perfect lighting, perfect water, perfect levels of carbon dioxide, etc. But it is entirely reasonable to expect to get to 50 to 60% photosynthetic efficiency. And when you think about what that means, when you move from 20% to 60% photosynthetic efficiency, that means you've just tripled the quantity of sugars that that plant is producing over the course of its entire lifetime. Does that mean you're going to produce triple the yield? Very seldom. In most cases not. You might get a 30% or a 50% yield bump, but you're not going to get triple the yield, either of plant biomass or of grain or whatever it is that you're harvesting and producing. So the question is, where does all the extra sugar go? It goes down into the root system and out through the roots as root exudates. And this is how we can build organic matter while you are growing a crop. You can build soil, you can regenerate soil health and build soil carbon levels while you are growing a corn crop, while you're growing tomatoes, while you're growing strawberries, while you're growing tree fruit. There should not be the necessity to have one crop that is extractive and another crop that replaces what that first crop extracted. This is inherently oxymoronic. So I became very inspired by the idea that these models of plant nutrition, these models of agronomy, they can address so many of our collective societal and ecological illness. With these regenerative agronomy models, we can greatly reduce and in many cases completely eliminate the need for pesticides. We can produce food as medicine, which is desperately needed from a public health perspective, particularly in North America. And we can regenerate soil health and we can regenerate ecosystem health at the same time. When I started having this conversation with growers about restoring soil health and growing food as medicine and reducing pesticide applications, growers were very intrigued by the conversation. They were very interested, and they expressed a lot of enthusiasm that these ideas make a lot of sense. It's something that they're interested in trying. But very few of them actually made the decision to make changes. So the ideas were interesting, but not enough to change behavior. And I realized if we want to facilitate significant change on scale, you have to offer an incentive. You achieve what you incentivize. And unlike what you have happening here in the UK recently, we do not have any significant government incentives for regeneration, for building soil health, et cetera. I mean, it's for cover crop seed cost share, federal program will help compensate and pay for seeds of up to $6 per acre. In other words, nothing. Those are some of the best programs that are available in North America. But I very much believe that you achieve what you incentivize. And so our approach to regenerative agronomy has been to obsessively focus on how do we deliver immediate economic results in the first year, in the first growing season. And this is an important distinction because for quite some time, the idea of regenerative agriculture was that there was a necessary transition period. You should expect to see yield declines, and that there was an initial investment that needed to be made in incorporating cover crops, incorporating livestock, incorporating different cultural management practices, and we should expect to have a future payoff in 3 or 5 or 7 years down the road from the current year's investment. Well, I don't know about Europe and the UK, but in North America, most farmers do not have a 3 to 5 year financial window. There needs to be an immediate economic crop response. And as a result, when we started thinking about how do we manage agronomy and how do we manage plant nutrition to produce an immediate crop response the first year, and I think that has been one of the foundational catalysts for why we have been so remarkably effective in scaling these models of agronomy across so many different crop types in many different regions. So I'm going to dive into a few of these pieces. So how many of you have observed crops where the yield was significantly disproportional to the quantity of fertilizer applied? We have this expectation that we need to apply so many units of phosphorus or potassium or nitrogen to grow a bushel of grain. How many of you have observed crops that significantly over-yielded the quantity of fertilizer applied that you expected? Okay, I see a hand. Okay, good. A couple dozen hands in the air. How many of you have observed crops that were resistant to diseases and insects when the presence of those diseases and insects was known to be in close geographical proximity? Okay, a number of you have, awesome. But many more of you have not. I realized after conversations yesterday that accessing SAP analysis has been more logistically challenging. It's funny because we use a lab in the Netherlands, Nova Crop Control, and all of our samples are exported from the US to Nova Crop Control for lab analysis, hundreds of samples per week, thousands in peak weeks. And it appears that it is easier for us to access the Netherlands from a customs perspective than it is for you here in the UK. So with that caveat and with that developing understanding of your difficulties from a logistical and border perspective, how many of you have used SAP analysis in the last 12 or 24 months? Okay, a number of you, oh, quite a few of you have. Wow. All right. So I'm going to skip here. The realization, the foundational piece of regenerative agronomy is that remarkable crop results are a reflection of these two pieces, managing nutrition and the soil microbiome. And you know what is not included on this list? I did not say that if you want to transition to regenerative agriculture, you need to eliminate pesticides and plant cover crops. Those happen after. You have to earn the right to reduce pesticide applications if you want to do so in a manner that produces immediate economic results. So what does regeneration look like? I described this cycle, this regenerative cycle. The first step of the cycle is increasing photosynthesis. This is foundational. I'll get into a few of the concepts that I developed over the years, but when you increase photosynthetic volume from a baseline of 15 to 20% up to 45 or 60%, you've tripled the number of sugars that are being produced every day. Those sugars are then sent out through the root system as root exudates to feed soil biology. So that's step 2. Then that translates to accelerated microbial activity, an increased microbial population in the rhizosphere and in the bulk soil environment, which then translates to these minerals extracting and etching minerals from the soil mineral matrix and making them available to plants more efficiently, which leads to step 5, increased disease resistance and insect resistance and exceptional harvest quality. The beauty of this system and of this cycle is that this is a self-perpetuating system. We have observed many growers who we work intensely with their nutrition management, agronomic management over the course of the first several years, but then as we begin to get the ecosystem pieces right, and we have the right companion plants, the right cover crop species, the appropriate nutrition management, this becomes a self-perpetuating system that doesn't require constant spoon feeding and constant support. I'm a very strong champion and advocate of using foliar applications, using seed treatments and so forth. We'll get into a little bit of that. But the long-range expectation should be that we develop regenerative agriculture ecosystems that are not dependent on constant inputs unless, and this is an important caveat, unless we have a cropping system or a soil management system type that requires it. If we are growing strawberries in the same soil year after year, then we have to make some allowances and consideration that that is not an ecologically diverse system. So as we have the regenerative cycle, we also have a degenerative cycle. That is the exact same process, except in reverse. We now have decreased photosynthetic volume, which leads to limited sugars going out through the root system as root exudates, which leads to poor microbial activity, which leads to reduced nutrient absorption from the soil profile, which leads to increased pest susceptibility, insect disease susceptibility, and reduced harvest quality and reduced yields. The only difference between a regenerative cycle and a degenerative cycle is you, the farm manager. Both of these cycles are self-perpetuating. The only difference in the cycle that will dominate in your cropping system is going to be dependent on the management decisions that you make. So in North America, we have the growing popularity of these 6 principles of soil health: understanding context, using cover crops, integrating livestock, etc. But here is where it is so easy. First of all, I agree with these foundational principles. But these foundational principles are not a recipe. This is not a recipe that you can simply apply universally across all cropping systems. And this is, I think, where the way that we need to think about this is if there are some aspects of these foundational principles that we cannot readily incorporate into our crop management system, perhaps because of food safety concerns, we can't incorporate livestock. Perhaps because of the cropping system, we cannot use cover crops. There are many different farm-specific and context-specific situations where some of these principles cannot be implemented. And we actually find that we can be remarkably successful in these situations, but we then have to compensate for what we are not able to include. There have to be other compensations that are made. If we cannot incorporate livestock, perhaps that means we need to grow plants with more aggressive root systems as cover crops and build our soil. We have to build soil microbial populations in some way. If it's not with livestock, then it perhaps might be using other tools. So that's the point I wanted to make is that these are important foundational principles. And if you necessarily need to miss one or multiple of them, you need to compensate and replace them in other ways. So this is a concept, the concept of the plant health pyramid is a concept that I first developed in 2011 and have discussed it extensively. What we observed, I have extensive information about the plant health pyramid available online if you want to dig. Actually, let me ask a question. How many of you have not heard the concept of the plant health pyramid, have not heard me talk about it? Okay, many of you have not. So I'm going to give you a very rapid overview, and if you want to dig deeper, there is an abundance of information available on this concept online. I would recommend you look at YouTube. We have a webinar series on the plant health pyramid that is readily accessible. This concept, this diagram, came about as a result of us working with many different growers in different contexts, different crops, very different environments, but observing many interesting parallels in how plants became resistant to different groups of diseases and insects based on what was happening inside the plant with plant physiology. So the foundational level of the plant health pyramid is when plants have complete photosynthesis. And actually, I'm recalling now, I believe in one of my follow-up presentations tomorrow, I'm going to be going through this, or maybe it's this afternoon, I forget, the presentation on increasing disease and insect resistance. I will be diving into this in more detail. So level 1 is when you increase photosynthesis, plants produce both a larger quantity and higher quality of sugars, and they become resistant to many of the soil-borne diseases. So soil-borne diseases like Fusarium and Pythium and Rhizoctonia, Verticillium, etc. The second stage is when they begin synthesizing, metabolizing nitrogen and synthesizing complete proteins very rapidly. And in plant sap, we see that they have presence of 0 nitrates and 0 ammonium levels. And all the nitrogen compounds that have been absorbed have been immediately converted to complete proteins. This gives the plant resistance to all of the various groups of insects with simpler digestive systems, particularly all the larval insects such as cabbage looper, European corn borer, corn earworm, tomato hornworm, etc. Also, aphids would be in this category. And at this stage, so these first 2 groups of resistance, these first 2 stages, are what we refer to as passive immunity. What has happened is that the plant has become so healthy and its nutritional profile has changed. The amino acid profile and the carbohydrate profile has changed to such a degree that you've removed the food source for the insects. The insects and the diseases are dependent on very particular carbohydrate profiles, very particular amino acid profiles, and when you change the plant physiological profile, you simply remove the food source. So this is what I refer to as passive immunity. The level 3 is when plants develop a surplus of energy. They have more energy than they need to sustain themselves and to successfully reproduce, and they begin storing this surplus energy in the form of lipids, plant fats, and oils. And we see this expressed visually in the plant. We can observe this when we have this glossy, waxy sheen on the leaf surface. And at this stage, many plants become resistant to many of the airborne diseases, mildews, rusts, bacterial infections, etc. And then the 4th stage occurs in parallel to a degree with the 3rd stage, and this is the stage at which plants actively upregulate their inherent immune system, the ISR and the SAR pathways. And at this stage, plants become resistant also to the beetle family, to Japanese beetles, Colorado potato beetles, flea beetles, etc. And through the active synthesis of immune compounds, plants produce this broad range of immune compounds, phenolics, terpenes, bioflavonoids, etc. This is a large group, tens of thousands of different compounds, hundreds of thousands actually, that in plain English we could just simply call essential oils. All plants produce these compounds, but the concentration of these compounds within plants can vary by orders of magnitude. And at this stage, plants shift from passive immunity to active immunity. Many of these compounds have very potent antiviral, antibacterial, antifungal properties, as well as being digestion inhibitors for the digestive systems of many insects. So, how do you achieve results immediately, consistently, and reliably? This has been the foundation of why we've been able to have the impact on the number of acres and number of growers that we work with. You cannot guess. You cannot guess. With many of the farmers, growers that we work with, we work with many large-scale growers as well as smaller-scale producers. And when you are making agronomic recommendations and decisions on crops that are worth hundreds of thousands, or in many cases millions of dollars, you had better not screw it up. You had better not screw it up. And I think this is my sense. I can speak to my own experience in the North American context. My perspective is that mainstream agronomy and some who are in what I'll call the alternative or regenerative agronomy space play entirely too fast and loose with making recommendations that they don't have in-depth experience with. And that, or maybe saying it a better way, is that they don't have a great deal of confidence, can be and is almost certain to be successful. So we have this mantra that I repeat frequently with our team and publicly: don't guess about whatever it is possible to measure. And so this is, when we begin working with a farm, we conduct a very thorough evaluation. This is just a quick snapshot of some of the laboratory assays that we use. But the laboratory assays are just one piece of the greater whole. We also conduct a very thorough evaluation to understand what are the disease pressures, what are the specific diseases that are problematic, what are specific insects, what are the dominant weed populations, because all of those things tell us a story. We have now been able, over the last decade and a half, we've been able to correlate specific diseases or groups of diseases with and insects with particular nutritional profiles. So I can say with certainty and with complete confidence that if you tell me that you have flea beetles on rapeseed, you will have deficiencies of either magnesium or sulfur or molybdenum or boron or some combination of the four. I don't need a laboratory test to tell me that, because I can have complete certainty that your plants are not metabolizing nitrogen and proteins properly as a result of an imbalance of those four elements. And when you address those four elements well, flea beetles are going to cease to be a problem. They're gone, they're done. The same is true of bean weevils, same is true of many other insect pests. If you have, well, I'm guessing with your climate, you may not have significant challenges with spider mites, powdery mildew, many diseases we can correspond to particular nutritional profile, and we know what is happening within that soil and plant ecosystem as a reflection of disease and insect pressure. So the laboratory analysis that we use, we're just beginning to use genomics analysis of the microbiome. We don't yet have a lot of data and experience with that. Then we use the second test that I've listed here on soil analysis is total digestion nutrient analysis. I don't really have the time to get into this, but it is important once we begin using biological nutrition management systems, our soils are capable of delivering much greater quantities of nutrition than what will commonly be described on a standard CEC analysis, either a MELEC-3 or ammonium acetate extraction. And we need to understand what our parent geological bedrock material is and what nutrients it is capable of delivering. Because there is this, I'm less familiar with the local context, but in North America there has been this idea, this mantra, that when all we have to do is we have to regenerate soil biology. And when we regenerate soil biology, the biology will provide all the nutrients that the crop requires. Well, sometimes maybe yes, if the parent geological material has those minerals available that can be released. But if you have a soil geological profile that contains no selenium, and that contains no molybdenum, and that contains no cobalt, it doesn't matter how abundant your microbial population is, they will never deliver those nutrients until you address them. Then the plant analysis that we use, we also use, primarily rely on sap analysis, and in the future, we're expecting to use genomics analysis of the microbiome that is inside the leaf and on the leaf surface. How many of you have heard of the rhizophagy cycle and are familiar with this concept? All right, a few of you are, many are not. This is incredible. I'm going to run short on the clock here if I keep diving into these topics. So, 20 years ago, as I was studying plant nutrition from an immune perspective, I came across this body of research, of agronomic research, that was trying to understand and describe how plants were absorbing nutrients in a context of soil biology. 10 miles from where I live, we have where the glacier dropped a lot of the rock, its rock burden when the glaciers melted. And so we have these large rock piles, and there are trees growing out of the rocks. There is no soil present. There has never been soil present. There are trees growing right directly on bare, flat rock surfaces, and these trees are normal size, normal age. A few of them have been cut open as an experiment to demonstrate, and the growth rings are exactly the same size as trees that are growing in soil. There are no soluble ion nutrients in that system. Mainstream agronomy for the last century or longer has subscribed to the notion that the primary means and the primary mechanism of plant absorption of nutrients is that plants absorb simple ions from the soil solution, that we have calcium, sodium, potassium, nitrate ions soluble in a water solution in the soil, and that the plant has the ability to absorb those ions and utilizes those ions as its primary source of nutrition. And it's true that plants do have this ability. After all, this is how hydroponics works. And if we rely on this model of plant nutrition, in an outdoor agriculture environment, we are relying on a glorified hydroponic model. That's it. It's that simple. There are two foundational problems with relying on agronomy that is dependent on water-soluble nutrition. The first problem is what happens when you have excess of water, and the second problem is what happens when you don't have water. You get nutrient leaching and a lack of nutrient availability in both of those situations. A few years ago, I was delighted to discover the work of Dr. James White and colleagues. Dr. James White is at Rutgers University. He first popularized the term, and he first described this concept of the rhizophagy cycle in the peer-reviewed literature 17 years ago, and it has remained largely unknown. But what he described is that as plant root tips are growing through the soil, the apex, the root cap, is actually quite open and porous and can absorb large quantities of bacteria. And we now understand that there are also fungal endophytes, but for the moment, I'm going to focus the conversation on bacterial endophytes. So these These are bacteria which can be recruited from the soil microbial community and actually live inside the plant, hence the term endophytes. Endophytes refers to an organism that lives inside the plant. They colonize the entire plant architecture. They are in the seeds, in the fruit, leaves. They completely colonize the plant. But as in this, at the growing root tip, the plant is absorbing large quantities of bacteria, and the first few inches immediately behind the growing root tip, these bacteria are reacted upon with oxidizing, in an oxidizing environment with reactive oxygen species, nitrous oxide, and so forth, that strips the cell membranes off the bacterial cells. And then these bacterial cells, some of which have been stripped, but not all, move through the plant's vascular tissue to cells and are actually absorbed into the plant cells through a process called endocytosis, and the plant then uses all the nutrients that are contained inside the bacterial cell as a form of nutrition, as a form of energy, but they don't have to disassemble them to a large degree. They will use bacterial proteins, bacterial peptides, and carbohydrate structures directly without fully breaking them down. But then a few inches back from the growing root tip, some of these bacteria begin to congregate. And as they begin to accumulate, they trigger the formation of root hair. Root hair begin extending away from the root, and these bacteria move out of through the root hair back into the soil environment, and the plant also releases carbohydrates and mucilage compounds that allow these naked cells that don't have a cell membrane to quickly reform their cell membranes. But in this process of the plants absorbing bacteria from the soil, moving them into the root system, and then back out through the root hair, the plant communicates with chemical signals and with electromagnetic signals to the microorganisms exactly what its nutritional requirements are. It tells them, hey, I need more zinc, I need more manganese, I need more phosphorus. The bacteria then move out through the root hair, and once they are back into the soil rhizosphere environment, they communicate that signal through quorum sensing to the rest of the soil microbiome. 4 minutes after the bacteria leave the these root hair, distant biology at distant mycelial networks, distances of yards away from the plant, immediately, selectively begin etching minerals out of the soil mineral matrix, precisely what the crop signaled that it required. And then it channels bacteria that contain those high concentrations of those minerals, bacteria that contain high concentrations of zinc or manganese or phosphorus, into the fungal mycelial network of mycorrhizal and saprophytic fungi, and channels those bacteria back to the plants. And in a matter of as little as 4 hours after the release of the signal of what the plant requires, bacteria carrying high concentrations of those particular nutrients will arrive back at the plant. Isn't that incredible? Plants have this symbiotic relationship. This is not particularly a symbiotic framing, but we could say that plants are farming bacteria. Plants are farming the microbiome, utilizing the microbiome as their digestive system. By the way, this also means that plants are not vegetarians. So this is an incredibly beautiful process. And we are still trying to determine fully and with complete accuracy what proportion of a cultivated crop's nutritional requirements can be supplied with this rhizophagy cycle. I'm of the persuasion that for most crops, the answer is 100%. And that's an opinion that is a reflection of a lot of experience and a lot of observation. But if you want to shut this process down, if you want to prevent the rhizophagy cycle from working, there are several very easy things that you can do to prevent this cycle from working. You can apply fungicides. That will certainly destroy it. You can apply soluble nitrogen fertilizer, and you can apply soluble phosphorus fertilizer. Each of those 3 tools are the 3 most effective tools that we know of for shutting down the rhizophagy cycle. Does that sound like modern agronomy or what? So, a few additional thoughts that I want to, and by the way, I'll also be diving into the rhizophagy cycle deeper into more detail and describing how how we then, in this context, with this understanding, how do we then manage nitrogen effectively, and how do we manage phosphorus effectively in one of my presentations later. We need to be thinking of seeds as vectors for microbial diversity. One of the challenges that we have is that many of our soils are not cycling carbon very well. Many of our soils are not sequestering carbon very well because of the lack of plant species diversity that we grow on those soils. We can have conversations. One of the tools that we use a great deal in our consulting work at Advancing Eco-Agriculture is using microbial inoculants, because we have observed microbial inoculants, when they are well designed and properly applied, to be remarkably effective and consistently effective. And I know that our experience is different from many people's experience when using compost tea and compost extracts and so forth. And microbial inoculants, I believe, are a very valuable and necessary and powerful tool. But we should be thinking about seeds as microbial inoculants. A corn seed contains 9 billion, with a B, microorganisms inside of it. And all plant species contain these, they have this associated microbiome with endophytes that lives inside the seed. And the microbiomes of all these plants, particularly between various plant groups, there's a great deal of diversity between plant groups. So when you think about the various grass families of warm season grasses and cool season grasses and the daisy family, cereal grains, there is a great deal of diversity. There is a smaller range of diversity between oats and barley and wheat. There's a much greater range of diversity or contrast between wheat and corn, or between wheat and corn and thistles. And many of our agricultural ecosystems suffer today. The soils are not able, the rhizophagy cycle is not able to function well because we don't have enough species diversity in our overall cropping system. There can be effects that carry over from year to year, but if we simply grow canola and wheat, that is not a rotation. There is not enough microbial diversity in that system to really regenerate soils very well. What we have observed is that when we begin working with growers to facilitate a transition, we find that the applications which produce the greatest economic crop response and are the easiest to apply are seed treatments. Mineral seed treatments and microbial seed treatments. And we have more information on our website at advancingecoag.com if you want to dig deeper into the recommendations of exactly what we're doing and how we're doing it. The application that can build soil health faster than any other is foliar applications. And there isn't even a close comparison. There is nothing else that comes close to a well-designed foliar application. And the key part of this phrase is well-designed. The historical expectation has been that when we use foliar applications produce a yield response at the expense of soil health, and that foliar applications simply allow plants to extract nutrients from the soil more efficiently, and that foliar applications are the fastest way to deplete soil health. And that can occur. That is a reflection of the design of how the foliar application was designed. So a foliar application needs to, its primary purpose, its primary objective needs to be to increase the plant's photosynthetic rate. And the way we've commonly been framed or taught to think about foliar applications is that the primary purpose of a foliar application is to address nutrient deficiencies. We pull a sap sample or we look at soil analysis and we anticipate that our plants are low in manganese or iron or phosphorus or nitrogen, and we put on a foliar application to address those nutrient deficiencies. When we do that, then it is reasonable to expect that those plants, now that those nutrient deficiencies have been addressed, and assuming we've identified them correctly, that those types of applications will allow plants to extract nutrients from the soil more efficiently than ever before and not really replace them effectively. However, if you design a foliar application with the intent of increasing the plant's photosynthesis, remember I said the threshold is a common baseline of 15 to 20%, you can easily and readily jump that from 20% to 40 to 50% with a single foliar application. It is a temporary spike. That spike can last, it is the length of time that this increased photosynthetic spike remains present is a reflection of the overall ecosystem health, overall soil health, overall plant health. It can last for as long as 4 to 6 weeks, or it can collapse in as little as 4 to 5 days. But you can have this temporary photosynthetic spike from a well-designed foliar application. And I would suggest that in many agricultural ecosystems, what we've observed is that this spike lasts for a duration of 2 to 3 weeks, sometimes longer. When you think about, and I have many examples of, or several examples of what this looks like on farms that we've worked on the blog, but when you have a well-designed foliar application, and that increases the quantity of sugars, depending on which crop that we're talking about. I'll use wheat as an example. A wheat crop that moves from 20% to 60% photosynthetic efficiency represents an additional, I think roughly an additional 150 to 180 pounds of sugars per acre per 24-hour photo period. So you think about what I just said, 150 pounds of sugar per acre per day. You cannot economically afford to buy enough sugar and to add that to the soil to compare with what that crop is able to produce with its photosynthetic engine. So a well-designed foliar application spikes and increases the plant's photosynthesis, which results in more sugar added to the soil to build soil biology, to build soil carbon. This is the single fastest and most economical way that I know of rapidly regenerating soil health, is to harness the plant's photosynthetic engine to trigger it and to accelerate it to rapidly produce more carbon than the baseline that we are most familiar with. This realization has been important for us. Most disease and insect susceptibility is a result of over-application of nutrients or application at the wrong time. If I could eliminate all nitrogen and potassium applications until 45 to 60 days post-planting on most crops, it would greatly reduce disease and insect pressure, and it would, here's the absolute surprise, it would increase yields on most crops, including wheat and canola. Putting on fertilizer applications at planting or before planting is an excellent mechanism to sell lots of fungicides and insecticides and not good for much else. So yield drag from overnutrient application is often greater than yield drag from water imbalances, from excess of water or from not enough water. I'll be having a book signing tomorrow, assuming the books actually showed up here. We'll see how many they are and how long they last. I'm around here for the next 2 days, and I'm looking forward to having conversations with you. Here's the various places that I'll be speaking and presenting. And you can learn more about the work that we're doing these various places. I would particularly suggest the second, my blog platform, johnkempf.com. You can find that right here. If you want to scan that with your cameras, that will take you to a link directly to the blog. So with that, I want to say thank you. Thank you for being here at Groundswell. Thank you for all the work that you do. And I look forward to having conversations with you over the next couple of days. Thank you much.